Good morning to all. So yesterday we left with bulk segregation analysis, or segregation analysis (BSA), where we could find that this pattern of bands, if it is persistent, that means presence of band in parent and absence of band in another parent, according to the type of the marker present. This is the pattern we get of dominant marker. So by this analysis, we can know that this band might be associated with the trait or linked with the trait. But from this, we cannot decide the linkage distance. We cannot know how far this particular polymorphism marker is located from our gene of interest. So for that, we perform linkage mapping, where we decide the distance in centimole, distance between the polymorphism, that is a marker, and the gene of interest. So for that, after conducting BSA, we isolate DNA from each F2 individual. If you have 200 individual from where you have bulk, let us say 15 resistant and 15 susceptible individual, then we isolate DNA for mapping, for linkage mapping. We isolate DNA from all these 200 individuals. And we genotype all these individuals for that polymorphic marker for which you have found the bending pattern and probable association. So we genotype all these 200 F2 individuals with that marker. That means we try to know that each individual, which kind of band is present. And we also phenotype each F2 individual, resistant and susceptible reaction. And we compare this genotype of F2 individuals with parent. That means each individual is classified as parental type or recombinant type depending upon the marker profile. I will tell you in the next slide how we classify that. So we classify them as parental and recombinant, and we calculate recombination fraction. That means number of recombinants divided by total number of individuals studied. And then we estimate the linkage distance by utilizing the mapping functions like Helden and Kozambi. So mapping functions are utilized to convert recombination frequency into distance in terms of centimole. So this is how we classify the individuals, segregating individuals as parent or recombinant type. So by genotyping this all 200 F2 individual after bulk segregation analysis, we classify the individuals. Now here let us assume that this E, locus E, is responsible for our trait. That is our gene of interest. Let us say E is responsible for disease resistance, small e is responsible for susceptibility to the disease. And the marker of polymorphism we are getting in bulk segregation analysis is locus F. This is the marker polymorphism. They are linked. So if they are closely associated with each other, then there are less chances of recombination between them. There might be recombination, but at very low frequency. When this low frequency recombination occurs, when you classify the individuals as parental and recombinant, you see the dominant gene of the trait and the marker. Let us say capital F means presence of band and small f means absence of band. Let us just consider the dominant marker. So with the resistant character for that particular F2 individual, here F is present. So this is called parental type because in resistant parent, this is the profile. Some of the individuals might be carrying susceptibility to the disease and they are expressing absence of marker. So this is also parental type, similar to parent 2. Now, when recombination occurs between marker and gene of interest, we may find recombinant types at low frequency. Here in this case, recombination frequency is 10%, 10 out of 100. And these recombinant types, if you look at this, then you can see here that the resistant to the disease, that is phenotypically observed. This is not profile at molecular level. This is phenotypically observed. So resistant to the disease, here it is present with absence of marker. So that indicates the recombination because now they are differing from parental type. And in another recombinant types, absence of the this susceptibility to disease is associated with, or we find susceptibility to disease with presence of marker. So this is also recombinant. So now you can see here recombination frequency is 10%, 5 plus 5. So it indicates that there's a 10 centimorgan distance between the gene of interest and the marker locus, which we have observed during bulk segregation analysis also. So this is how linkage mapping is carried out. But still the distance of 10 centimorgan is too much. Generally, we need a marker which is tightly linked with the gene of interest. So we expect that the marker distance from the gene of interest is 1 centimorgan or less than 1 centimorgan. So you must find only 1% recombinance here rather than the 10, but we can say it is loosely linked marker. If following the independent assortment rule, if the recombination frequency is 50%, then it is not linked to the gene of interest. The marker may be present on another chromosome, a polymorphism may be present on the same chromosome, but here the polymorphism is so far from the gene of interest. If we get 50% recombination, suppose, just to have idea, then marker is said to be unlinked to the gene of interest. Now we may have got another marker, another polymorphism during bulk segregation analysis. This is just to explain what do we mean by loosely linked marker further away from the gene of interest. So here capital G and small g, this is, let us say, our gene of interest. This is responsible for disease resistance, small g is responsible for susceptibility, and this is the polymorphism, marker polymorphism. So here also we can isolate DNA from all F2 individuals, and we genotype this individual with the marker locus, whether the band is present or band is absent. And we also phenotype the F2 individual for the disease reaction. And then we, similar to the previous case, we classify the individuals as parental and recombinant. But now you can see here H is far away from the G, so many recombination events are possible between them. And because of that, we'll find higher number of recombinants. Here it is 40%. So distance is 40 centimorgans. So it is loosely linked marker, or roughly it is very close to the unlinked marker. So we try to find out these kind of the markers which are much more close to the gene. Theoretically, the distance must be 1 centimorgan or less than that for effective marker assisted selection. So this is how linkage mapping is carried out. Once you get the recombination frequency, the recombination frequency is converted into linkage distance by the mapping function. So the normal function which we utilize is Morgan's function. That means if there are 10% recombinant individual, then we say distance is 10 centimorgan. Morgan's function assumes additivity of the distances and zero interference. Morgan's function says that there is zero interference. I hope you are clear with interference. Interference is a phenomenon where one crossing over event and one locus is affected by the crossing over event at neighboring locus. So Morgan's function assumes that there is additivity between the distances and there is no interference. Additivity means the distance between E and F, suppose if it is 10 centimorgan, there is another locus, let us say Z here, and here distance is 20 centimorgan, then the distance between E and Z would be 30 centimorgan. It means 10 plus 20. So additivity of the distance. That's the Morgan's function. Haldane's function assumes that there is non-additivity. That means you cannot add the distances directly. There is non-additivity between the distances. So here in Haldane's function, 10 plus 20 may not be 30. It may be something else than 30. That's the meaning of non-additivity. But Haldane's function assumes that there is no interference. Interference is absent. So this is the formula. You can convert recombination frequency here, fraction, into the distance. So if let us say recombination frequency is 20%, in decimal terms it is 0.02. So we'll keep here 0.02, 20% means 0.2, sorry, 0.2, and you will calculate the distance. Kozami's function is most widely utilized function for QTL analysis and marker mapping. It assumes non-additivity, and it also assumes that interference is present. And this is the formula for Kozambi. R is again recombination fraction. So if there is a 10 centimorgan or the 10% recombination frequency, then we put here 0.1, and accordingly the distance will be calculated. Now, when the genes are closely situated or markers are closely situated, Halden's and Morgan's function can be utilized. Mostly Halden's function is utilized. But when you are deciding the distance between the polymorphisms which are far away from each other, then interference may play the role, and non-additivity may comes into picture. So for whole genome marker mapping across the genome, across the chromosomes, Kozambi's function is utilized. Kozambi is Indian scientist. He discovered this function. He came out with this function around 1945, before India's independence. Now this function is most widely used in QTL analysis. These are the applications of marker state selection. You can incorporate markers in any breeding program, theoretically. Backcross, recurrent selection, any breeding program, pedigree, population improvement, any program. So we can use marker state selection for biotic stress resistant breeding, abiotic stress resistant breeding, marker state backcrossing, marker state recurrent selection. Molecular markers can be utilized in many ways, like here for identification of restorers also molecular markers can be useful. Like suppose to identify the restorer, the conventional technique is you cross all the genotypes with your CMS lines and you evaluate hybrids. Suppose you have 400 germplasm lines, then to identify the restorer you have to cross all these 400 with your CMS lines. So there will be 400 hybrids. You have to study pollen fertility in the hybrids. Whichever genotype is able to restore fertility in F1, F1s are fertile, then you say that that germplasm is restorer. And wherever you get pollen sterility in F1, you say that genotype is non-restorer. So that's the conventional way. But if you can identify the marker which is linked with the restorer gene, then by molecular profiling only, without going to the F1, you can reduce the number of germplasm to be studied. Suppose you have 4,000 germplasm. You can screen this with marker which is linked with restorer gene, and then you can reduce your search to merely 15 to 20 germplasm. And then further you have to confirm the restorability. That means you need to make the cross between this 15 to 20 confirm lines with utilizing the markers. Now you will be crossing this 15 to 20 genotypes with your CMS line. You will evaluate F1. That is to be done. But now you have reduced number of crosses to be handled from 400 or 4,000 to only 15 to 20. So this is one just example how we can utilize markers in plant breeding. We use markers for hybrid purity check. We can use for hybrid vigor analysis, for improvement of the quality trait also, for marker assisted gene pyramiding. Gene pyramiding is a concept where we incorporate race specific resistance genes in one genotype. So we can use markers in that also. These are the applications of marker. So now we'll deal with quantitative trait loci analysis, QTL analysis. So these are the questions which are asked during QTL analysis, and we get answers after performing QTL analysis or QTL mapping. How many genes influence the expression of the quantitative trait? You get to know after QTL analysis that how many loci are responsible for particular quantitative trait. So a quantitative trait may be governed by 10 loci or 15 loci. But remember that when we say 15 loci are governing the quantitative trait, it means there are 20 alleles present in diploid individuals. 10 loci, suppose, then there are 20 alleles. So you have to have that picture in your mind. Now we also get to know what is the level of influence of each gene. Now quantitative traits are governed by polygenes. Polygenes means the genes which are having small, similar, and cumulative effect according to Mathur. But so far what we have known is, suppose 10 loci are responsible for inheritance of particular quantitative trait. They are contributing to the quantitative trait. Then each locus might have a different influence on the trait. Some locus may have major influence of the trait. Other loci might have minor influence on the trait. So that's how polygenes are somewhat different from the QTL. QTL term was coined by Gelderman. Now, where these genes are located on the chromosome, that also we get idea, because the markers we have utilized to be associated with particular quantitative trait, we know the positions of those markers on chromosome. We already know, because we also construct molecular maps by utilizing during the QTL analysis. So that's how we know the placement of the marker. If the marker which is already placed on 5th chromosome, if it is associated with particular quantitative locus or loci, then we already know the placement of that marker on the 5th chromosome. Let us say on shorter arm, we'll be able to know that linked QTL is located on shorter arm of 5th chromosome. So we also get to know the location of the markers or QTLs on the chromosome. Which gene action is responsible for the trait? That also can be known, whether it is additive, dominance, epistatic, we can know by QTL analysis. Which alleles have favorable effects? Because when we say marker polymorphism, as I told you earlier, there are marker alleles. One allele of the marker may be associated with positive allele, contributing positively towards the trait. Another marker allele may be linked with a recessive allele, that is negative influence, recessive allele, which has negative influence on the quantitative trait. So out of, let us say, 10 loci and 20 alleles, you can classify these 20 alleles as positive and negative depending upon their effect on the phenotype. And we can also know the function of each gene to the trait, that how each allele and each gene is contributing to the quantitative trait through central dogma, through gene expression, which proteins are encoded by these genes, and how particular allele has positive influence on the trait, another allele has negative influence, and how their protein products are contributing to the trait. The most difficult part to answer is this, the last question. And not only the proteins directly contribute to the genes, there might be interaction between those proteins. We know that as epistasis or gene interactions. So there the complexity arises. But at some level we can do this also. So QTL analysis, let us just first recall the properties of quantitative traits. They show continuous variation. You see bell-shaped curve when you analyze the whole population for a particular quantitative trait. They are highly influenced by the environment. These are the traits. So there might be any of such quantitative trait you are dealing with. Yield is very complex trait. Quality. So I'm not talking about the qualitative trait. Qualitative trait means purple flower, white flower. There are examples of qualitative traits. Quality means protein quality, oil quality. These qualities, nutritional qualities, those qualities might be governed by many loci. So in that term, in that sense, quality traits may be governed by quantitative loci. So what is QTL? The loci governing the quantitative trait. Simply you can put it like this. The loci governing the quantitative trait or contributing to the quantitative trait, they are known as QTL, quantitative trait loci. And what is QTL mapping or QTL analysis? QTL mapping, we are trying to identify many markers which are linked to these loci distributed across the genome. So at a time you might be dealing with any of such quantitative trait. Yes, the QTL term was coined by Gelderman in 1975. So these are the properties of quantitative traits governed by many genes. They are measurable. We utilize metric units to measure them. They are highly influenced by the environment. Characters are not easily observed because of the continuous variation. You cannot have distinct classes here. Like in quantitative traits, we can have purple flower and white flower. Distinct classes are possible. But in QTL, to measure the quantitative trait, distinct classes are not possible because the continuous variation is present. In QTL analysis, we try to identify markers, polymorphisms, which are linked with the loci governing quantitative trait. So we are not directly identifying the locus, but we are identifying the marker which is linked with a particular locus, or many markers which are linked with many loci governing the quantitative trait. That's the basic idea. So we know about DNA markers. This is just recalling their basic the basic concept of DNA markers. They are signs and flags which indicates the presence of particular genotype or phenotype. This is other way of explaining monomorphic and polymorphic marker. So this is our ladder sequence. Let us say these are the 4 genotypes. This bending profile indicates monomorphic pattern. So you cannot distinguish individuals on the basis of that. But this is the SSR profiling of these genotypes. So this SSR allele is common in all these individuals. So we cannot distinguish this individual. But on the basis of this profiling, you can distinguish one individual from another. This is codominant marker. This is a dominant marker. This is monomorphic band. You cannot distinguish the individual, but here band is present, here it is absent. So on the basis of that, you can distinguish this individual. So dominant marker will show presence or absence of band. Linkage mapping, I already explained earlier after bulk segregation analysis, but there I was talking about deciding the mapping distance between one single polymorphism locus or marker locus and the gene of interest. But now we are extending our search to many loci, many markers linked with many loci contributing to the quantitative rate. So if you use many markers distributed across the genome, then the chances are higher that we'll get the markers which are linked with the gene of interest. The basic idea is same, crossing over frequency and linkage. So genes and marker which are closely or tightly linked with particular QTL, they will be transferring with that QTL from parents to offspring. One allele of the marker may be tightly linked with the positive allele of the QTL, and another allele of the marker may be linked with negative allele of that QTL. And recombination frequency and mapping distances are utilized to analyze these results. So this I already explained. So same concept is utilized, but it is extended to many loci rather than limiting to the single locus. So these are the steps of QTL analysis. There are main 3 steps. We produce mapping population from 2 diverse parents. So the parents must be diverse to be utilized for QTL analysis. More diverse the parent, there will be more allelic diversity for the loci governing the quantity trait. Suppose your character is pores per plant, then you have to select from your germplasm, try to select the parent which is having highest number of pores per plant, and you select another parent which is having the lowest number of pores per plant. Like that, for any quantitative trait, you must select the diverse parent. Not only on the basis of phenotype, that means pores per plant or plant height, disease resistance or susceptibility, you might be dealing with any quantitative trait. But parents' diversity must also be analyzed at molecular level. By utilizing various marker techniques, you must also assess the diversity of parents at genetic level, DNA level also, not only at phenotypic level. So that way you can select the diverse genotypes. And these 2 diverse parents showing the maximum diversity, and they are crossed to create this mapping population. Then we identify the polymorphism. Now from that selected parent, we identify maximum number of polymorphic loci. More the loci you identify, that's better. So generally 100 markers are good, minimum 100 loci you must be, polymorphic loci you need for QTL analysis. Maximum number, there is no limitation. There is no limitation. But as many as possible polymorphic loci should be used for QTL analysis. Then we analyze linkage between these polymorphic markers and any QTL. So that's the way you identify the marker loci which are linked with the QTLs. Out of so many polymorphisms you have detected between 2 parents, you identify the marker loci. Now mapping populations. So as I told you earlier, recombinant inbred lines are most widely utilized mapping population because the recombination frequency is higher. Many recombination events have taken place during their development from F1 meiosis onwards up to F8 or F10 generation. The RILs are most widely utilized by parental mapping population for QTL analysis. Double haploids are also utilized, and nowadays you may also see the papers where backcross population or backcross F2s have been utilized for QTL analysis. We can also utilize F2, F3, F4 populations for QTL analysis. However, RILs are better because of their homozygosity, because of the normal segregation of 1:1, because of more recombination events. More and more recombination events you consider, you get fine map of your QTL. That means you get to know the tightly linked markers. More and more closely situated markers can be identified, or fine mapping is possible by RILs. But nowadays, we also perform joint linkage and association analysis, where we utilize linkage as well as linkage disequilibrium for mapping of the trait or gene mapping. We'll come to that point later on after QTL analysis. And for that, name and magic populations are utilized. I will describe this population later on. And once you develop you start developing mapping population, at the same time, you also identify the polymorphism present between these two diverse parents, and number of markers required is also dependent upon the genome size. In diploid species like rice, you may require less number of markers, but in polyploids or the species with higher genome size like wheat, hexaploid wheat, you need more number of markers because the genome size is huge. So it depends upon ploidy level or genome size. So I already explained this, that you select parent on the basis of phenotypic diversity as well as genetic diversity. Now, once the polymorphic markers have been identified in parents, we only utilize those polymorphic markers for genotyping of individuals of mapping population. Suppose you have developed 400 RILs from those two parents, then we'll genotype all these 400 RILs for their genetic constitution. Now, once you have developed RIL, they will show 1:1 segregation for any marker locus. 50% of the RIL, particular allele of the marker will be present, and 50% another 50% RIL, another allele of that marker would be present, or that marker locus would be present. So we genotype these RILs for presence of marker allele, which marker allele is present. So this is for one marker locus. We do the same for all the marker loci simultaneously. So we genotype this individual. Now genotyping method might be any. You may utilize PCR-based marker like SSR. You may utilize genotyping by sequencing concept, utilizing next generation sequencing. You may utilize this SNP markers, utilizing these NGS methods. We'll come to that later on. So we genotype 400 RILs developed from those two parents. We phenotype this RIL also for the trait under consideration. So suppose the trait under consideration is ports per plant, then you will measure the ports per plant for each RIL. And this data, genotyping data and phenotypic data, are incorporated in the software as input, and finally the analysis is done. So when you genotype the mapping population individuals of different mapping population, so just I have kept this slide so we can have an idea that what we are doing by genotyping. So if the marker is codominant, and these are the two parents, for that marker we'll have two alleles, capital A, small a. The parent 1 is homozygous for this allele. Parent 2 is, of course, we utilize highly homozygous parents for development of mapping population. There should not be heterozygosity in the parent for any marker locus. Then we cannot utilize those. Parents must be homozygous. The mapping population individually you are studying may be heterozygous, but highly homozygous parents should be utilized. But QTL analysis and association mapping is also possible in natural populations, so that is another aspect. But right now I will stick to this homozygous parent and utilizing linkage for QTL analysis. Now F1 will show both the alleles because we are talking about codominant marker. So if we have developed F2 population from that F1, this is the F2 individual. For simplicity, only 12 individuals are considered here. So some of the F2 individuals might be homozygous for this allele. Some of the individuals are heterozygous, and other individuals might be homozygous A, small a, similar to P2. So this would be the profile of codominant marker in F2 generation. If you use the codominant marker in backcross, then again there are two alleles for particular marker. This is the situation in F1, and if you have developed backcross, then in backcross we know that there will be one heterozygous, a 50% heterozygous plant and 50% homozygous plant. There is 1:1 segregation. Some of the individuals will express this allele P2. Some of the individuals are heterozygous. So the profile itself can give you idea that here F1 was crossed with P2, because the backcross progenies are expressing homozygosity close to P2, similar to P2, and heterozygous situation. So this would be the profile of backcross progenies if it is codominant marker. If we consider RILs and double haploids, where the segregation is 1:1, and all the individuals are almost homozygous, 100% homozygous in double haploid, and almost homozygous in RIL, up to F6 or F8 RILs we are talking about. So in codominant marker, both the parents are expressing this allele. F1 will be heterozygous, so it is codominant marker, so it will express both these alleles. RILs are homozygous, so you don't find heterozygous RIL. The first RIL is similar to P1, considering the molecular profile. Second is similar to P2, like that. Like that, we classify the RILs, alleles present in the RIL, and roughly you will find 1:1 segregation. Like you see, almost there are 6 alleles, marker alleles from P1, and there are 6 allele present in P2, 50-50%. So 50% RILs are expressing this allele, 50% RILs are expressing the alleles from P2. So you'll find this 1:1 segregation for a particular marker locus. And that's why markers are always qualitative in nature. And in QTL analysis, we'll see how this qualitative markers can be utilized to assess quantitative traits. Now the same populations, but what happens when you utilize dominant marker? The dominant marker shows presence or absence of the band. When you analyze F1 DNA sample with that marker, you will see presence of band. Definitely in F1 band must be present, because F1 will receive this allele as well as another allele from both the parent. And then this is the profile of F2 individual. Now the band would be present in homozygous individual, band would be present in heterozygous individual also, that is 75% proportion of F2. Only 25% individual will show absence of band. So you will find more bands here, and band will also segregate according to 3:1 pattern. So 75% of the F2 individuals will show presence of band, and 25% will show absence of band. If we utilize dominant marker in backcross generation, then band is present in P1, absent in P2, is present in F1. And when you subject backcross progenies to this dominant marker, in backcross the segregation pattern is 1:1. So nearly in half of the progenies band would be present, half of the progenies bands are absent. So you can compare these two for a dominant marker. We find more bands present here as compared, because the segregation is 3:1 for presence of band and absence of band. And here the segregation is 1:1, one for presence of band, one for absence of band. And we can similarly analyze RILs and double haploids with dominant marker. So you can see here in F2 the patterns are different, and codominant markers are more effective than dominant marker. In backcross and RIL and double haploid, where segregation is 1:1, dominant and codominant markers have equal. efficiency. They have equal efficiency for backcross and RILs. Now, in RILs, we'll again find 1:1 segregation. So if it is dominant marker, it is present in P1, absent in P2, and F1, the band would be present. Now, if you look at this pattern, there is 1:1 segregation. 50% RIL or double haploid individuals, band is present, and 50% the band is absent. This is for single marker locus. Still, we are not talking about linkage. We are just talking about this concept is very similar to monohybrid cross of Mendel. We are focusing on one marker locus and its two alleles. So expected ratio for single marker locus in F2 for codominant marker is 1:2:1. So here 1:2:1. That pattern will, 25% will be like this, 25% will be like this, similar to P2, and 50% individuals will show both the alleles after molecular profiling. Backcross will show 1:1 ratio for codominant as well as dominant marker. Recombinant and the double haploids, all this population will also show 1:1 segregation pattern. And whether the marker is showing the segregation or not, that can be judged by chi-square test. The similar chi-square test we apply to find out whether both the alleles are segregating normally or not in F2 population, where we perform chi-square analysis with the expected and observed frequencies. You can say after that the alleles are segregating normally. I mean, there is no segregation distortion, there is no meiotic drive. Both the alleles of a marker are segregating normally in 1:1 fashion. Now, if our crop is polyploid, then identifying polymorphic marker will be more complicated, because for single locus there will be 6 alleles in polyploid crop if it is hexaploid. Like in wheat, there will be 6 alleles for a single locus. So it is much more difficult to create marker map in polyploids, but in some of the polyploid species where diploid parental species are available, evolutionary parents are available, then we can use the marker data from diploid species to generate marker map in the evolved hexaploid species. But again, there is a problem with some of the crop species where diploid relative parents or diploid relatives are not available. In that case, it will be much more difficult. Now in QTL analysis, we judge the significance of the marker by load value. So what we have gone through up to this point, I just want to recall that we have identified 2 diverse parents. We have developed mapping population, like RIL, let us say. We have identified polymorphism between these 2 parents. And utilizing those polymorphic markers, we have genotype or mapping population individuals. So we have genotype and phenotypic data of parents and mapping population individuals. We have those data. Now we subject those data, genotypic and phenotypic data of mapping population to the software. Generally, MapMaker software is utilized. So subject those data to the software, and then the load value is calculated. So load value is just the ratio of linkage to no linkage. It is also called logarithm of odds. So it is again related with error probability, and more the load score, it is better. Load score of 3 indicates, because it is based on logarithm, load score of 3 indicates there is a chance of error is 1 out of 1,000. That means in 1 out of 1,000 case, there might be no linkage and we are detecting the link. So this is related with alpha error or type 1 error of statistic. So load score indicates the significance of particular marker contributing to the quantitative trait. So we can fix some threshold value here. Generally, you find the threshold value of 2.5 to 3. If the load value of 3 indicates the error rate is 1 out of 1,000. So this is the threshold value. When you associate the marker loci with the quantitative trait, the data of quantitative trait you have incorporated from that mapping population individual, let us say pods per plant. You have measured pods per plant from all these RILs and you incorporate the data, then you see this peak. This peak indicates that marker loci are associated with the trait. phenotypic value of the trait. But this peak, how to know whether it is due to chance factor or there is a real association? So for that, we are utilizing the load value. So you can see here this peak is going beyond this load value. So if this is the threshold value of 3, then it might have 6 or 7, value of 6 or 7. It indicates that error probability is very low, and the linkage we have detected between this marker loci and the trait, it is significant. It is highly significant. We'll come to this later on. Now, so far, as I mentioned earlier, we have genotyped the parents with markers. So these are, let us say, we consider, for simple understanding, we consider 4 markers at a time. And through this slide, I want to show you how molecular marker maps are prepared during QTL analysis. So 4 markers are there: marker E, F, G, and H. This is the profile for the parents, A and B. A and B are the alleles of the marker. These 2 are the alleles of marker E, a separate locus. These are the 2 alleles of marker F. Alleles here means the band size. They differ. And by looking at this picture, you might guess, you might be guessing it. This is codominant marker because both the alleles are expressed. Sizes are also different. Let us say these are the 20 RILs obtained from these 2 parents. And we are genotyping these RILs with this this particular marker, all 4 markers. This is the genotypic profile of those RILs with marker E. So you can see here in some of the individual, A allele of marker E is present. In some of the individual, B allele of marker is present. So this is for one marker. There is another marker, marker F, allelic profile, A and B alleles of marker. Now, I want to categorize these RILs as parental type for these 2 markers and as recombinant type, so that I can decide the distance between these 2 marker loci. So now we are talking about 2 marker loci and 4 alleles. So let's just look at this first RIL, the profile of first RIL. The profile for both this marker is exactly similar to P1. So we classify that as parental type. This profile is similar exactly to the P1 profile for both the marker. So it is also parental. This profile of third RIL, both the bands are similar to parent 2 for E and F. So this is also parental type. But if you look at this fourth RIL, this allele belongs to marker E, another allele belonging to marker F. This profile is different from both the parent. None of the parent has this profile. So we classify that as a recombinant RIL. This is also recombinant RIL number 12. You can see here the profiles are different from parent. That doesn't belong to one parent. Shows the characteristic of both parents. So we classify that as recombinant. And that's how number of recombinant individuals are identified from the marker profile E and F. So once you have the recombinant number, you can calculate the mapping distance. And that is also observed in the scoring with AB scoring pattern. And this is marker E and F. So you can see here AA, that means it is common to P1. This second RIL is also common to P1 here, as we have observed. Third RIL is parental type similar to P2 because P2 is expressing B allele for both the marker, the same profile we observe here. But RIL number 4, you see it is BA, so this is recombinant. AA parental, BB parental, so these are all parental up to this point. RIL number 12 is recombinant. So here you can see A, B is there. So likewise, the scoring is performed and number of recombinant RIL for markers are decided. And then we get the distance in terms of centimorgan utilizing the recombination frequency, where recombination fraction is the number of recombinant divided by total number of individuals, and you get the distance between the markers. Once this molecular marker map is prepared, here I have shown for 4 markers only. So likewise, as we have considered for E and F, the same way we can also work out distance between F and G. But the first RIL is parental type. The second RIL here is recombinant. because the profiles are similar to both the parents. One allele is similar to P1, another allele is similar to P2. So this is recombinant. So likewise, and if you want to just observe from this profile, if you want to observe the recombinants for F and G, then I have given the colors. You can observe that. Purple circle indicates that that's a recombinant for G and H. Likewise, the recombinant individuals are identified for all marker loci, and we can prepare the molecular marker map. Along with that data, marker data, we have also given the phenotypic data to the software. So now the software will associate this marker map data with the trait value, and that's how we'll be able to know whether this marker is linked with the QTL or not. So first we prepare molecular marker map, and then we associate this marker map with the quantitative data. So whatever polymorphisms we are utilizing, it is better if the polymorphism is distributed across the genome. But this thing nowadays it is under control, but before 10 to 15 years it was not in our control. I mean, you just identify the polymorphisms. You might have 400 to 500 polymorphic loci identified. You might have prepared marker maps, but you don't know whether all these markers are distributed across the genome randomly, or many of these markers are concentrated on few chromosomes. Now, it is better if these polymorphic loci are distributed across the genome, because chances of getting the link marker increases if the markers are distributed across the genome. Why? As I told you earlier, because the loci governing quantitative trait may not be clustered together always. Suppose 10 loci are responsible for the trait, 4 or 5 may be linked, and other 5 may be distributed across the chromosomes on separate chromosomes. So if your polymorphisms you are utilizing for QTL analysis, if they are distributed across the genome, then chances of getting linked marker increases. But generally this distribution is not random, it is non-random. That means it is biased. Sometimes the polymorphic loci you have identified might be limited to one chromosome or few chromosomes only, and that will create a problem. So the whole chromosome itself is one linkage group. So when you perform molecular marker map in rice, if you are performing molecular mapping, then you will find maximum 11 linkage group. If all the markers are distributed to all the chromosome, that means the number of chromosome present in the gamete. If it is hexaploid wheat, then you will find 21 linkage groups, because the whole chromosome is the entire linkage group, the whole linkage group. So I already told you this, we utilize Poisson-B mapping function most widely. Sometimes in some of the paper, Haldane function is also utilized. Poisson-B is preferred because it takes into consideration non-additivity and interference growth. And as you are mapping the markers on whole chromosome, your distance is too wide. There are chances of interference and non-additivity. So to prepare the molecular marker map across the chromosome and genome, we use Poisson-B function. But the recombination frequency will vary. Linkage mapping distance is based on recombination frequency. And even if Poisson-B function is utilized, we assume that there is a linear relationship between recombination frequency and distance. But it is not so if you consider the crossing over events occurring within the genome between the chromosomes, homologous chromosomes. Because there are some hotspot for recombination where recombination is frequently taking place, but there are cold spots also where recombination very rarely takes place, like the region around centromere. So if there are two loci flanking, they are flanking the centromere, they are situated on both sides of centromere. And now within that region, there is rarely recombination occurring. Now you would find a very much less recombination frequency between these two loci. So you will say that they are linked. Actually, they are very much far farther away from each other. But because it is a cold spot for recombination, you said that they are linked. So such problems are there when you perform molecular marker mapping, when you look at the markers on particular chromosomes. So these are the issues we need to have in our mind. Now, how these qualitative markers can be utilized to assess quantitative trait. So mostly, as I told you earlier, RILs are utilized for analysis, and F2, F3, F4 population can also be used. Any molecular marker technique can be utilized for this polymorphism. Generally, SNPs are preferred. They are most widely utilized nowadays, but SSR can also be utilized. I suggest that I will also give you the paper of bulk segregation analysis by Michael Moore. So I will mail you that paper. So you go through that paper and you try to read 2 to 3 papers on QTL analysis, foreign authors paper. So you will get more idea about QTL analysis, what is done and how the data are represented in the paper. So if I give you basic idea of QTL analysis or how the markers are associated with the phenotypic trait, then suppose you can consider any trait. Here there is example of cattle, milk yield in cattle, but let us consider quotes per plant. And you are analyzing F3 or F4 individuals or RILs. So let us say you are analyzing RILs and you have 400 RILs from 2 diverse parents for ports per plant. So when you genotype these RILs and when you arrange this RIL in particular order, descending order, from the RIL giving the highest number of ports to the lowest number of ports, if you analyze the marker profile, then some bands you observe when you are considering the RILs with higher number of ports per plant. But as you proceed from the RILs showing the highest number of or higher number of ports per plant towards the lower number of ports per plant, you see that certain bands will disappear. That indicates that these disappearing bands are associated with higher number of ports per plant. And as you proceed towards the RIL having a lower number of ports per plant, certain bands will appear, new bands will appear. That indicates that these newly appearing bands are associated with lower number of ports per plant. So this is the basic concept. Software will do that. We are not going to do that because there might be 200 to 400 RILs at a time with you, and there might be 5,000 to 10,000 markers. So that's a huge data to be handled. MapMaker program will do this. So this is the general concept I explained. A minimum size of the mapping population for QTL analysis is 200 individuals. Not less than that. If it is less than that, you will not get good publication. So it has to be 200 or more than 200. Because if the number of individuals are only 10 to 15, then we are not taking into account all the recombination events between those markers, and the whole molecular marker map would be mistaken. So 200 individuals are needed to take into account the recombination events and mapping the markers on chromosomes and deciding the distances between them. So mapping populations can be utilized. Any mapping population we can use, but RILs are most widely used. MAGIC and NAME are used for combined linkage and association analysis. We'll come to that later on. NILs can be used. Normally, NILs are not used for QTL analysis, but if you are focusing on major QTL or clustered QTL, then near isogenic lines may be used. So these are the steps. I have already gone through these steps. So. Now, this is the basic idea of QTL analysis, how qualitative markers can be utilized to assess quantitative trait. So, if our markers cover larger portion of the genome, then there will be good chance that there will be a marker which is linked with a particular QTL locus. Now, if the gene is linked with the marker locus is linked with the particular QTL locus, I would call it gene. If it is linked, then One allele of the marker will always be linked or associated with positive allele, and another allele is associated with negative allele. So presence of that allele, which is linked with the positive allele of the QTL, presence of that allele will always be associated with higher number of ports per plant. Suppose if that is the character. And presence of another allele linked with the negative allele of the QTL, whenever that allele is present, you will always see lower number of ports per plant. If these genes and markers are segregating, if they are farther apart, then you will not see this kind of association. That means positive allele of the QTL, there are equal chance to be present with presence of any allele of the marker, and negative allele of the QTL, there are equal chances. So I'm talking about independent assortment. If there is independent assortment, you will not find this relationship. So utilizing this logic, this is how marker profile and phenotypic data are used to establish linkage. So let us talk about this marker E, and you have genotype 20 RILs with this marker. These are A and B are the alleles of this marker. So what software will do? We have incorporated all this data, presence of particular allele, presence of another allele to the software. So considering marker E and the trait, what software will do? Software will classify each RIL according to class A and class B, according to the alleles of the marker present. Here I have shown EB, that is the B allele of marker E, and EA, that is A allele of marker E. Now software will classify these RILs into two groups. All the RILs showing fragment A and all the RILs showing fragment B. Now we have also incorporated phenotypic data. So software will calculate the average number of ports per plant for all these RILs grouped under category A. So this is the mean of that RIL grouping. And it will also work out the average number of ports per plant for all these RILs grouped under class B. This is the mean. Now, if there is a significant difference between these mean values, that means this marker is linked with the QTL, governing number of ports per plant. Because the B allele of marker E is, if it is linked with positive allele, then you will always find higher number of ports per plant because it is favorable allele. Higher number of ports per plant in that group. It would be significantly higher than the another group where the A allele of marker is always linked with negative contributing allele of QTL locus. So you'll find the significant difference. And whenever we get significant difference between these two groups of RILs, the software will conclude that marker E is linked with the one QTL governing number of ports per plant. That way we may identify many markers linked with other quantitative trait loci governing number of ports per plant. Now let us talk about another marker. There is marker H. Again, we have two alleles, A allele and B alleles of marker H. And again, software will classify the RILs according to marker A and marker B category. So there are again two groups. But now this marker H is so much farther away from this QTL locus. So there is independent assortment. There are many recombination events possible between this marker locus H and QTL. So they are away. I mean, the marker is away from the QTL and there is independent assortment. That means the positive allele of the QTL may be equally present with HB or HA. A negative allele may be equally present with HB and HA. So in that case, if you work out the mean performance of these RILs, because the presence of particular allele is not always associated with the favorable effect, or presence of this allele is not always associated with negative effect due to independent assortment between marker and the QTL, now the mean difference between these two groups would be non-significant. Remember that the RILs are grouped according to marker class, not according to the QTL, only according to the marker. If there is a linkage, we'll find a significant difference between these two groups. If there is no linkage because of the independent assortment, every allele has equal chance to be with positive or negative allele. This grouping, once it is done, we will not find a significant mean difference between these two groups. That indicates this marker H is segregating from the QTL and it is not linked with the QTL. So this is our molecular marker data. Utilizing the concept of linkage on the basis of this grouping and phenotypic trait, we can map marker with quantitative trait. So this is the scoring system. This is how we incorporate our data to the software. This is the profile for parent A. So we may be giving the score of A. This is B marker. That means these are the two alleles of one marker locus. Heterozygous individual expressing both the alleles may be given the code H. There are other codes also, C and D. C means the individual is not homozygous for AA. If the individual is not homozygous for A, the individual could be homozygous BB or heterozygous. Remember, here A and B are alleles of one marker locus. Likewise, we may have the category D, which is not homozygous for allele B. That means either it is heterozygous or homozygous A, where there is no homozygous B present. And dash indicates missing value. So we have to analyze the profile for each marker. Suppose if you have 400 RILs and 200 markers, there will be 80,000 data points. You have to analyze each one and you have to incorporate in one Excel file. So this is how the Excel input file is prepared, where 1 indicates homozygous A, let us say 2 indicates heterozygosity, and 3 indicates homozygous B, and dash value indicates missing data. So there are other data present in this Excel file. These are the markers. These are the SSR markers. There could be SNP markers also. And this way we have the number RILs. There might be 200 to 400 RILs. These are the number of individuals present in mapping population. These are the number of markers utilized. Generally, these are higher than 200. Markers may be more than 100. It is better if you utilize more and more number of markers. But again, you have to take into consideration the material to be handled, resources available and the funding available. These are the classes. 1 means A, 2 means heterozygous, 3 means homozygous B, C means 4. So these notations are given. You can tell the software. You can change these notations accordingly, but you should convey the meaning properly. Dash means missing value. And apart from that Excel file, we also, in the same file, we also give the phenotypic data of RIL. Suppose the trait is ports per plant, we give that data to RIL. And this is just a snapshot of that MapMaker program. We have to run this program in command base. And if possible, I will also demonstrate how we can use this MapMaker program in one lecture. Then we incorporate all this data input file, and this is how the distances are worked out between various SSR markers. And when this profile is, output is given to this MapChart 2.2 software, then we generate the marker map data. You can see the SSR markers have been placed on the linkage groups according to the recombination frequency between them. And then this marker profile is associated with the phenotypic trait for QTL analysis. This is just construction of marker maps, molecular marker maps. So that's all for today. Tomorrow I will discuss other methods used in QTL analysis, like interval mapping, single marker analysis, and all other relevant information to QTL analysis. So I end this lecture here. If you have any question, then we can deal with the question. Okay, so if there are no questions, then I end our session here. Again, we'll tomorrow from 11 to 12, we'll continue with QTL analysis.